On OsteoBites today, we are talking with Dr. Lillian Guenther from Dana-Farber Cancer Institute and Harvard Medical School about utilizing CRISPR screening to identify novel genomic targets for osteosarcoma. OsteoBites webinar and podcast present the latest in osteosarcoma treatment, research, innovation, and hope. Welcome to OsteoBites, everybody. It is such a gorgeous day, and this requires a great snack, which involves coffee, of course, and going old school with some fig Newtons. Today, we're talking with Dr. Lillian Guenther from Dana-Farber Cancer Institute and Harvard Medical School. Dr. Guenther will be talking about utilizing CRISPR screening to identify novel genomic targets for osteosarcoma. So excited about this topic. Dr. Guenther received her bachelor degree from Brown University and her MD from SUNY Downstate College of Medicine. She spent 1 year as a Howard Hughes Medical Institute NIH Research Scholar studying osteosarcoma in Dr. Lee Helman's laboratory at the National Cancer Institute. Lily is currently an instructor in pediatrics at Harvard Medical School and performs research in the lab of Dr. Kimberly Stegmaier. She cares for pediatric cancer patients at Boston Children's Hospital and in the Jimmy Fund Clinic at Dana-Farber Cancer Institute. Dr. Guenther was a finalist for our OutSmarting Osteosarcoma Grant. We just wish we had more money because our group of osteosarcoma families and scientific advisory team found her research to be truly hopeful and innovative. Our panel today includes 2021 MIB Junior Board member Matt Allen, and MIB Junior Board President Maeve Smart, and Amy Woodcheck, a physician's assistant and childhood cancer survivor. I'm your host, Anne Graham, osteo warrior and executive director for MIB Agents. MIB Agents is dedicated to making it better for our community of patients, caregivers, doctors, and researchers, with the goal of less toxic, more effective treatments and a cure for osteosarcoma. We do this through direct patient and family support, through funding research, and through educational programs and resources. There is so much to do, and we need your help to do it. If you can walk, run, or bike, we ask that you do it for kids with cancer. Recruit your friends, create a team, challenge your coworkers, run solo, just whatever it is, just do it to make it better. You can participate anytime from June 1st to August 31st. It's virtual. You can do your 5K, half marathon, or full marathon, or a combination of all of them. You can't do your marathon in 3 months, though. You got to do each thing all at once. You can do it on a treadmill or a Peloton as well. Those count. We are grateful for your effort in advance in this, and we are also grateful for the sponsorship of BTG Specialty Pharmaceuticals. Together, we can make it better. Please register at mibagents.org/outrunning. While you jot that down, Dr. Guenther, would you get us started by introducing yourself, please?
Hi, my name's Lily Guenther. I'm a junior faculty member, as Anne said, at Dana-Farber, and I just want to thank you so much for having me today. It's really exciting to be presenting to this group, and I'm excited to share our work with you on that is trying to make it better for osteosarcoma patients in the lab. Yeah, thanks so much.
My name is Maeve. I am a 2-time osteosarcoma survivor. I was diagnosed in 2011 and 2014, and now almost 7 years NED. I'm a pre-med student at Northeastern University in Boston, and as Anne mentioned, I'm honored to be serving as the MIB Junior Board President this year. I'm also currently interning at the Janeway Lab at Dana-Farber, and I'm very excited to be back as a panelist today on OsteoBites, so thank you for having me.
Hi everyone, I'm Matthew and I'm a member of the MIB Junior Board, and I'm also involved with MIB Gamer Agents for about the last 2 years. And basically what we do there, just for some background, is play video games with children who are either in treatment or their family members, just so they can get a break and be able to do something that they enjoy. And just some other things about me. I'm a second-year student in a college, and yeah, that's me basically.
I am Amy Woodchuck. I am a physician assistant and childhood cancer survivor. I have special interest in osteosarcoma and have been active with the MIB program since its first Factor conference.
And thank you, Anne, and thank you everyone at MIB. I am really excited to be here and to meet all of you panelists and talk to everyone on this call today about our work utilizing CRISPR screening to identify novel targets for osteosarcoma. I absolutely don't need any introduction to this disease on this call, but we all know and we're all passionate about the fact that osteosarcoma really needs new treatments. And that the fact that we have not moved the needle in this disease in multiple decades. And in particular, a major focus of these efforts has been on metastatic disease, as that is excruciatingly difficult to cure. And you can see, in this representative survival graph below that patients with metastatic disease really do exceptionally poorly when they present up front. And this is also true for kids who relapse with osteosarcoma. And so, we know that this is a very heterogeneous disease and that it has a lot of challenging features, some of that which I'm going to get into today. We need to approach this with different strategies than we have in the past. So I am a member of the Stegmaier lab at Dana-Farber, and the Stegmaier lab's focus is on multiple different types of cancer. I'm very proud to be the osteosarcoma lab leader in the Stegmaier lab. The Stegmaier lab was not really working on osteo before I joined, but we've changed that. But in multiple different types of cancer, so Ewing sarcoma, another bone tumor, leukemia, neuroblastoma, we're using an approach that is called the Pediatric Cancer Dependency Map to try to identify unique features about different types or subtypes of pediatric tumors that can be utilized for novel treatment strategies. So, by this, there's a lot of different efforts that go into this pediatric dependency map process. One of them is this bucket that is genetic or chemical perturbations. In this bucket, we find CRISPR screening, which I'm going to talk about today, which is a genetic modality to actually edit out genes across the genomes in cancer cells. We also apply chemical library screening to all of these cell lines that go through the dependency map screening process. We're also looking very carefully at all of these different pediatric cancer cell line models genetically and molecularly. So using whole exome sequencing and now more specifically whole genome sequencing, which is particularly important in osteosarcoma, as well as RNA sequencing, epigenetic profiling, and other modalities to really get at the inherent characteristics of each of these models. And then using advanced computational analysis to really generate what are specific modalities that we could use to target individual types of pediatric cancer. So for example, osteosarcoma. And let's see, so let me go ahead. So there have been many cancer cell lines screened in this process. To date, more than 700 cancer cell lines across adult and pediatrics have been profiled. So this is a very powerful map that can allow us to really hone in on features that are specific to individual cancers, just based on the global picture we get of the total N of cancer cell lines we have. But this graph represents really the pediatric lines that we're talking about that are in the pediatrics dependency map. And obviously osteosarcoma is our focus today. And I'm very proud to say that, since I joined the Stegmaier lab in 2015, we've doubled the number of models that we have that have been screened in this way, really providing more power to the data that we have. And we have multiple more lines that are active and still in the process of being screened as we speak. This includes a lot of low passage PDX or patient-derived xenograft-derived models, which are a different type of model that can help us get at the heterogeneity of osteosarcoma, which I'll talk about. And we also have a process through the Broad Institute, which is called the Cancer Cell Line Factory, that's generating lines that are very close to patient samples. So now we have a couple of lines that are being profiled that have been validated using this process, which is also very exciting. So I like to use this cartoon to kind of explain what CRISPR screening is, because that's what I'm going to be talking about. So if you can imagine, if you have these 3 different genes, A, B, and C, and you have this Pac-Man that represents basically getting rid of or eating the function of that gene A. And what is the effect of that? So when gene A is destroyed or eaten up, these cells in yellow are just growing as they usually would, right? They're replicating, they're dividing, they're happy. And so that's not something we're particularly interested in terms of a CRISPR target. Then we have gene B, where when that gene is destroyed, you see the growth slows down a little bit. There's not as many cells there, but they're still growing pretty well. And then you have gene C, which when you delete that gene, you actually get either cell death or you just get no growth at all. And this is what we call a dependency gene. So it's a gene that we think when we target, for example, in an osteosarcoma cell line, and we can get rid of that gene's function, that disrupts significantly or causes cell death in those osteosarcoma cells. And that's what we want, and that's what we want to study. And this process, this genomic screening modality, has led to many, many different efforts that have actually gone as far as clinical trials, some of which the Stegmaier lab has been involved in, and other groups as well. But just to highlight some of the studies here for you, it's been a really powerful technique that really has led to things that are in clinical trials currently, including at Dana-Farber and other institutions. So it's an exciting way to really profile cancer cell line models and to better understand targets for individual diseases. But one of the questions that I had when I was starting to do this work was really, are osteosarcoma models, are these cell lines that we're putting through CRISPR screening, are they representative of patient disease? Because if they aren't, then, are we doing the right thing here? And, I think we're talking mainly about established cell lines when we're talking about osteosarcoma models, but I just want to point out that in osteosarcoma, we're very lucky that we have a lot of different types of models from which to draw from. We have patient-derived cell lines and xenografts, as I mentioned previously. We actually have genetically engineered mouse models that we can work with for osteosarcoma. And of course, we have dog or canine models. And so this is really important. And the reason why it's important to really sample a diverse number of models is because, we know based on, a couple of these sequencing papers that I've referenced here, but as well as others, that osteosarcoma is incredibly heterogeneous. This is a recent paper that came out looking at all different pediatric cancers and really profiling mutational burden and other features of those cancer cell lines. And osteosarcoma really ranks near the top of this left-hand column, which is all different types of pediatric cancers in terms of number of mutations that are found in the tumors themselves. So whereas many pediatric tumors we talk about as being quiet or having few mutations, osteosarcoma is one of the noisier types of pediatric cancer, and really is closer to some of the adult diseases that we see on the right. So this lends itself to some of this heterogeneity. Another example of this is this plot on the right that, for those who aren't familiar, it's called a circus plot, which basically shows the chromosomes present in an osteosarcoma cell around as a circle, and basically demonstrates through this, truly you can appreciate the sort of chaos of this. All the different lines connecting different sites across the chromosomes represent different inter and intrachromosomal events, as well as changes in copy number of individual genes and mutations as well. And this can be really this is incredibly diverse between patients. So really, when we were talking about heterogeneity, osteosarcoma is a unique beast. And significant efforts have been made through many different groups, including, some patient advocacy groups, including Teresa Beach's effort and the POWR group. And then this graph, which is was part of a presentation recently from the Osteosarcoma Target Initiative, looking at just ways of subdividing pediatric osteosarcoma tumors. So, this is something that's far from being systematized, and it's being talked about amongst many, many people who are thinking a lot about osteosarcoma, ways to subdivide this disease into more digestible groups within this heterogeneity that can be targeted individually. And one thing I want to point out, I'm going to come back to, is MYC-amplified disease, which is a subset of disease that I have an interest in, that is highly represented in osteosarcoma, and in particular in one of these subgroups proposed by Target. So one way we're looking at our models and our models that are going into CRISPR screening to try to understand really what we're dealing with in terms of how these relate to pediatric tumors is we're using something called CellLiner, which is a tool that was recently published in Nature Communications. And it's basically an unsupervised alignment method to look at RNA sequencing data across basically all available primary patient samples, patient-derived xenografts, and cell lines. And so just to give you two other disease examples before I go into osteosarcoma, you have this Ewing sarcoma little cohort at the top that I've circled in blue, and you can see how all of those tumors are really neatly, tucked in one specific spot, with the patient cell lines and the patient samples and the established cell lines really on top of each other with very similar gene expression. Another similar example is these peripheral nervous system tumors, which I've highlighted below in light blue, and these are mostly neuroblastomas, which again are very homogeneous. And now, where is osteosarcoma, you might ask? Well, it's all kind of in this middle mushy area amongst some fibroblastic tumors, amongst some soft tissue tumors, and you have some bone in there. And so I'm just going to kind of zoom in on this to kind of show you and appreciate what we see in this data. So all of the purple plus signs here represent primary patient samples. And then we have a couple of different types of PDXs that are included in this analysis. And then in blue are the established cell lines that have gone through CRISPR screening. And you can appreciate that there's this cloud at the bottom here that's tight, that has a lot of different primary patient samples that seems to have very even gene expression. And there are a lot of PDXs in there as well. And then you kind of have this sort of spread out, differentially expressing different genes group of cell lines and patient-derived xenografts and patient tumors. And you can see that many of our cell lines are up in here, which is not completely not representative of patient disease, because there are some primary patients in here, right? But you can appreciate why some people criticize some of these models for maybe not being completely faithful to patient disease. Well, what I'd like to point out is that we're trying very hard to increase the representation of cell lines and models that are going through DepMap that are in this cluster below. And so there are 3 cell lines here now. And when I started in the lab, there was only 1. And so we're gradually improving it and trying to improve the heterogeneity of what we're looking at. So I think what is the punchline of what I'm saying is that more models are probably better. And so one of my major efforts in the lab is to continue to access more cell lines, more patient-derived xenograft lines. And this has come from a really great collaboration we have with Alejandro Cordero at UCSF to incorporate his PDX-derived cell lines in our pipeline, as well as the Broad Cancer Cell Line Factory that I mentioned earlier in the talk. And just to show you, that these really do represent faithfully the tumors that they come from. This is a figure from Alejandro Cordero's lab's paper describing these models from Cancer Discovery a couple of years ago. And this is just showing you the correlation of copy number between the primary tumors down here on the x-axis y-axis, and these patient-derived models that have come from those primary patient tumors. And you can see that across each of these tumors, these are really red, meaning that there's really nice alignment between the primary tumor and the model that was made from it, suggesting that these are really great models to use. Another thing just to point out is that, osteosarcoma, because of its heterogeneity, I think a reasonable question to ask about CRISPR screening is whether or not CRISPR screening will identify anything actionable or anything, usable in osteosarcoma compared to these other more quiet tumors that I've already mentioned. And we recently published the spectrum of the pediatric cell lines in DepMap in Nature Genetics. And this is just a figure from that publication that shows you that actually osteosarcoma, which I've outlined here in red, has one of the fewer numbers of dependency genes. So those genes I told you about with the Pac-Man that really kills the cell function and makes the cells die, there are fewer of those in osteosarcoma models than there are in other pediatric cell lines, which are highlighted in these other colors, which suggests that yes, there are fewer events that we can actually look at using CRISPR in osteo compared to other tumors. And something else that has been, thought about a lot about in osteosarcoma and has, been criticized about looking at CRISPR screening in osteosarcoma is just thinking about the chromosomal copy number events in osteo. And you can see here that osteo has more copy number changes than many other different types of pediatric cancer. In fact, all that were profiled in this paper. But I just wanted to say that we have really advanced correctional techniques that go into the CRISPR screening that eliminate some of the cutting toxicity that we see from CRISPR using in areas where there's large copy number gains or losses. And so we do feel like that we're able to correct for that problem specifically. As to the first problem that I said, that there are fewer numbers of dependencies in osteo compared to other tumors, I've sort of taken a different approach to looking at osteosarcoma dependencies than in some other diseases. Instead of looking at individual genes that are dependencies or that are really important across all the cell lines in the specific disease, i.e. Ewing sarcoma, neuroblastoma, anything else, take your pick. What I've chosen to do instead is to look more systematically at classes of genes that are dependencies across osteosarcoma tumors. So this is basically one of the major things that we found using this type of analysis. So when we look at all the osteosarcoma cell lines that we have screened using CRISPR, we have found that different genes involved in different DNA damage pathways and DNA damage repair pathways are dependencies in individual osteosarcoma cell lines. So this is rather than looking at an individual gene that's a dependency across all lines, this is taking an approach that's more line by line and looking more individualistically, but getting out an appreciable pattern here, which is that DNA damage repair is probably quite important in osteosarcoma. And this may not be that surprising to many of you, given what we know about the heterogeneity of disease in osteosarcoma and the frank, number of copy number changes and chromosomal aberrations, et cetera, but was very interesting to us and to me in particular. And I've sort of thought about different ways that we can go from using from this analysis to identifying novel ways to target this disease. So the first that I want to mention and just share with you a little bit is a top target that we found that was enriched. So in a subset of osteosarcoma lines, we found that this gene SMARCAL1 is an enriched dependency. And I think you can appreciate that this is far and away one of the top enriched dependencies in osteosarcoma as opposed to all other cell lines. And each of these dots represents a gene. And so when we saw this, I was immediately interested in this for a number of reasons. reasons. SMARCAL1 is an enzyme. It's an ATP-dependent helicase enzyme that's important for the stability of DNA replication. In particular, it activates the DNA damage response pathways in response to stalled DNA replication during S phase, a phenomenon that's called replication stress. So this is a very, sort of busy diagram, but I just wanted you to appreciate that SMARCAL1 is sort of a guardian of the genome during DNA replication, and it's activated directly by ATR as well as indirectly by two important kinases we know that are involved in cell division and stability, WEE1 and CHEK1. And basically, when SMARCAL1 is involved, it's during a period where a replication fork, so where DNA is replicating, is stalled for any reason, and that could be for a number of different reasons. And SMARCAL1 comes in and basically stabilizes this single-stranded DNA and recruits a whole host of other proteins to then basically make this replication fork pause, regress, and then restart normal DNA replication by repairing damaged DNA. And one of the most important ways that it engages to do this is homologous recombination, so an important DNA damage response pathway. One reason that I was really excited about this gene is that we saw in the dependency data that it is a dependency that is uniquely associated with MYC amplification and osteosarcoma. And so, there are relatively few osteosarcoma cell lines that are included in this analysis. I told you that there were 10. There may have only been 9 actually when this diagram was made. But the red dots represent a MYC amplified osteosarcoma, and they really trend towards being more negative in a dependency score, meaning that this gene is much more critical in those cell lines that are MYC amplified. This is just shown in a different way on the right, where you can see by a linear regression analysis that these cell lines in red that are osteosarcoma have a trend towards having a MYC, a higher MYC expression than other cell lines that are not dependent on SMARCAL1. And you can see that for the rest of the cell lines in the analysis, and there are many other MYC amplified cell lines besides osteosarcomas, that this is not true. So we thought that this was really interesting because this is a unique feature of osteosarcoma. Why do we care? Well, I think many in this crowd know that MYC amplification affects probably about 1/4 to 1/3 of osteosarcoma tumors, and that this is a finding that has been now found in multiple retrospective analyses that's associated with poorer prognosis in osteosarcoma patients. And so this is an example from the target data that shows that MYC gain is associated with inferior survival, and this is another similar Kaplan-Meier curve from a cohort at Dana-Farber that shows that MYC amplification is associated with worse overall survival. And this is now hopefully going to be validated prospectively in an upcoming clinical trial of osteosarcoma patients. But overproduction of MYC basically leads to grossly dysregulated cell growth, increased DNA damage, and a whole host of other changes that can be related to just aggressive cancer. So the first thing that I did was I went ahead and validated this gene at low throughput to see if knocking out SMARCAL1 by CRISPR, as you can see on the left-hand side in a couple of different cell lines, we basically got rid of SMARCAL1 in the cells, and we were able to see that they grew inferiorly in short-term growth assays. The other method that we used was a method called the Dtag model, which I'm not going to really get into, but basically allows you to completely degrade by proteasomal degradation SMARCAL1 protein in the cells. And so if you just focus on the last line of this Western blot, see we've completely degraded SMARCAL1 and let these cells these osteosarcoma cells grow in culture for several weeks. And you can see that they have a markedly reduced number of colonies, suggesting that their viability is really impaired with the loss of this protein. I told you that SMARCA1 is phosphorylated and activated by Atr, and this is something that obviously was very interesting to me because Atr inhibitors are being used in the clinic for different cancer patients. What I found when I tested a bunch of different cell lines, osteosarcoma cell lines, was that if you sort these cell lines by SMARCA1 dependence, so you have several lines that are dependent in black as opposed to one line that's not dependent in red, you can see that there's a trend towards more sensitivity of these lines to a small molecule inhibitor of Atr, and that this in one representative cell line does decrease phospho-Atr as expected, and increases gamma H2AX, or the amount of DNA damage in the cells. So this was exciting, and I did observe a similar trend with a couple of other types of small molecule inhibitors that inhibit DNA damage response, which could potentially be exciting for clinical trial development in the future, given that this could be essentially a biomarker of increased response in this subset of tumors. The other thing, just to get back to the MYC connection for a second, is that we discovered that when we knocked out SMARCA1, that we actually had an increase in MYC levels in the cells. And we haven't fully got to the crux of this yet, but there is prior literature that suggests that SMARCA1 actually directly represses MYC activity, which is an interesting theory when applied to osteosarcoma, that perhaps MYC, which is creating all of this DNA damage, as I mentioned, in the cells, is being tempered by SMARCA1 expression, and that's why SMARCA1 is so important in these MYC amplified tumors, is to actually limit the amount of DNA damage that is perhaps occurring, and allow those cells to survive. So this is something that we are also exploring very actively in the lab. Another thing that's really exciting to me that I just wanted to mention to you, but is very early on, is that we have evidence now that SMARCA1 may be a potential germline predisposition gene in osteosarcoma. And so this paper on the left was recently published using a cohort of survivors from St. Jude. And they found that a little over 2% of their osteosarcoma patients have a predicted pathogenic or likely pathogenic, meaning a loss of function, heterozygous mutation in the germ lines of these patients with osteosarcoma. I subsequently approached Lisa Mirabella, who's an epidemiologist who focuses on osteosarcoma germline genetics at the NCI, and she and I are collaborating on this project now. And she has a large cohort that she recently published of more than 1,000 patients who were treated for osteosarcoma, and has germline sequencing on all those patients. And she similarly found, when she looked in her data, when I asked her, that a little over 2% or 2.5% of this more than 1,000 patients have a predicted loss of function mutation in the germline of SMARCA1. So this really suggests that this is a real signal, or something that should be investigated further as the potential cancer-causing gene in osteosarcoma. So this really highlights, I think, an interesting feature of this gene, which is that it has sort of dual roles. I've told you it's a critical dependency in some tumors, which means that it's present and it's needed in those tumors, but it's also a predisposition gene, meaning when it's lost, or at least partially lost, that it seems to lead to osteosarcoma in another group of patients. I think just highlighting the heterogeneity of DNA damage repair pathways in osteosarcoma, and just all that we have to learn about the intricacies of this disease. So this is just sort of a busy text slide, but we're working on lots of different features of this target in particular. Basically looking at differences in amounts of DNA damage and replication stress levels in the tumors that depend on SMARCA1 and don't, to try to learn something about the biology of these tumors, to really look at MYC amplification as potentially a biomarker of SMARCA1 dependency. And that could be really important for patients, because MYC amplification is something that is used now to classify patients with osteosarcoma, and may be able to guide for further clinical trials of potentially relevant small molecules, such as the DNA damage response inhibitors that I mentioned. And then working on this feature of SMARCA1 as a cancer dependency gene and a cancer predisposition gene rather in osteosarcoma, we're looking at this further, and actually going to start modeling this in a genetically engineered mouse model of osteosarcoma. So that will be exciting in the future. Some other things that I'm doing in the lab and that I'm interested in doing in the future as a result of the CRISPR screening data is obviously we're working on other targets that have screened for us in these tumors, but we're also doing a secondary in vivo screen to look at basically all the dependency genes that we found in osteosarcoma cell lines to see which of them, when we knock those same genes out in a mouse model of osteosarcoma, which are the most important genes in vivo, that is in a living organism. So to really get at what are the most important dependency genes that we should be going after that are perhaps more relevant to patient disease. And another project that I'm actively doing right now is following up hits from a genome scale screen similar to the one that has been done in all these other cancer cell lines to really look at what CRISPR knockouts are synergistic or have a greater effect when combined with PARP inhibitors, a class of DNA damage response inhibitors that are really of interest in osteosarcoma. And so we have some interesting targets there that we're also following up. But I just want to thank you guys for your attention and thank all of the members of the lab at Dana-Farber, as well as all my other collaborators and career mentors, some of which I mentioned during the talk, and my funding sources for this important research. And I'd love to take your questions.
Oh, great. Thank you very much, Lily. I enjoyed it thoroughly, and I'm happy I was able to make it this afternoon. So this was great. My question actually have to do with, for example, the relationship that you've identified between SMARCAL1 and MYC. And does that raise issues of that it's important to be looking at pathways more than individual genes? And how does this CRISPR technology enable you to do that or not?
Yeah, I mean, with the MYC connection, I mean, as I said, it's interesting because, we have the power of all these other cell lines that are not osteosarcoma. And so we can really see that this is an osteosarcoma specific phenomenon.
Mm-hmm.
And I think, just the highlighting the need for models is like, we need to increase the number of MYC amplified models in the pipeline to really, to test, to really understand if this is a biomarker. We're actually working on sort of manipulating MYC levels in the lab to see if that toggles SMARCAL1 dependency in the cell lines to suggest that it is a directly correlated relationship. But, so in terms of pathways, I think, the MYC amplified osteo is an important subset of osteo that we need to work on and make progress in. And so it's exciting to me that this is a potential avenue for those patients specifically. I think that MYC amplification probably lends itself to certain subsets of DNA damage, including replication stress. And that's certainly been shown in other published literature of other cancer types. And so that's something I'm particularly interested in as it relates to this target. I hope that sort of answers your question.
Yes, but in a more general way, I was just using your SMARCAL1 and MYC as a very important concrete example, but more the whole issue of pathway evaluation rather than individual genes.
Right.
And what your thoughts are about that.
Oh, absolutely. I think with the heterogeneity of osteosarcoma that it's going to be really important to look at line by line or small subsets of osteosarcoma models to be able to better delineate what's really driving those tumors in terms of dependencies that we can target, et cetera. I think that with 10 models, we're able to see this. I sort of showed you that RNA-seq data to make the point that we're really dealing with a very diverse entity here, and that we need to be able to better represent all those different types of disease that is osteosarcoma. But I think that taking that sort of more pathway level approach is going to lead us down more interesting paths than just to say this is a handful of genes that happen to score across all the different osteosarcoma models. I'm not sure that those might be interesting, but we haven't found anything particularly stunning by doing that type of analysis so far.
Right. Thank you.
Yeah.
So another question we had, Doctor, was Is the level of MYC amplification directly correlated to outcomes? And wondering if patients with low-level MYC amplification is considered the same as high-level MYC amplification.
Yeah, it's a great question. So I think that if you look at all the different ways that this has been looked at in patients retrospectively, meaning so taking a bunch of patients who were treated on a trial or through a certain avenue of sample collection and looking at them in the background, they've been looked at lots of different ways, and one of them is how many copies are actually present of MYC. I don't think we exactly know that answer yet. We do have some evidence in the Dana-Farber cohort that I showed you the Kaplan-Meier curve for, that increased copy number or copy number over 8 numbers of copies was the sort of cutoff for poorer prognosis. But I think a bigger sample cohort looking at this forward thinking will really answer that question. When we look at the cell line models of osteosarcoma, it's actually interesting. There's really not that much variety between the cell lines and the numbers of MYC copies. So we're dealing with somewhere between like 4 to 8 copies of MYC, which is not that much when you compare to something like MYC amplified neuroblastoma, for example, that has like 30 copies of NMYC. And so there's not that much variety within the models, and perhaps that will improve as well as we get more models screened.
Dr. Roberts?
Yeah, just a couple of comments. First, I'll just say also that we know that a lot of these changes are driven epigenetically, and there's lots of more ways to get MYC amplification per se than through genomic copy number amplification, right?
Yes.
And your SMARC data actually really intrigued me. We've got some data, hopefully it's coming out soon. If anyone's reviewing the paper, please be kind, that kind of suggests that metastasis actually requires downregulation of MYC. And so your data was kind of interesting in that driving MYC levels up, although it might create a more aggressive disease locally, it might actually prevent metastatic spread. But we can talk about that another day. I wanted to ask, so I was also intrigued by your RNA-seq data. I was really intrigued by your RNA-seq data. And I know my question is complicated, so follow me out here. But, doing screens in plastic is almost necessary because that's how you get high throughput, right? But we know that every cell changes dramatically once we put it on plastic.
Yeah.
And I wonder if you showed that really cool data that showed that there's kind of some cell lines that retain a lot of features similar to the patients and others that don't. Was there any relationship to the length of time that they'd been passaged, or why did some of them look that way? And did those that looked more like the patient samples, did they look more like their parent samples, like the tissue from the actual patient than others?
Yeah, so we know that the last part of what you asked is true, looking at Alejandro Cordero's models. So we know that his cell lines that he has derived, that his lab has derived, that now grow in plastic, are very similar when you look at the RNA-seq compared to the primary patient samples that they came from. I showed you the copy number correlation, but we also have RNA-seq. I've seen RNA-seq data from him that looks that way as well. In terms of the established cell lines that we've profiled in DepMap and whether or not there's a correlation to length of time in culture, et cetera, there does not seem to be, which is interesting. The cell lines that we've done a lot of work to try to say, is that cluster that's more enriched with primary patient samples, does that cluster represent more primary tumors as opposed to metastases? Does it represent, like trying to parse out the data from all the metadata we have associated with these treehouse and other samples to see if we can figure that out, or what we know about the cell lines, which is far from complete for some of them, but we haven't been able to really tease that out. One thing that we do notice is that that cluster that is greater has more like basic bone biology in its RNA-seq features. So it's more like, suggestive of like primitive to mature bone gene expression is popping out in those. So I got excited and I thought, oh, maybe the other ones are Mets, but they're not. Like we've tried to assess that before, and they're not. But that is one thing that we've found.
Do the dependency hits differ between the two groups, between the clusters?
Yes, somewhat. It is interesting. I think that we've only had the last couple of the 2 cell lines, 2 out of the 3 that are in that cluster. We've only had those in the dependency data for like the last couple months. And so we haven't done as an extensive analysis on this as we should, but we do think that some of the dependencies differ, and that's something that we're looking into more.
Thank you so much. Another question, it is 2 parts, but it looks like breast cancer might be similarly heterogeneous like osteosarcoma. Is this correct? And as well, is it possibly hopeful for osteo with a roadmap identified?
In terms of the first part of the question, I mean, yeah, I think it's breast cancer is I think there's a lot of heterogeneity of different types of cancer, I should say. I think that the advantage we have in breast cancer is that we have some subsets that we've been able to identify that are now actually even treated differently, right? Like hormone receptor positive versus negative. And we just haven't made it that far in osteo. And that has to do with a lot of things. It has to do with patient numbers. It has to do with funding for research, which we're working on. It has to, a lot of features and, lack of samples and just fewer samples, I think is a big one. But there's a lot more focus on this now. And so I think we'll be able to make more headway in osteo in that department.
There's been a family question wondering, aside from obvious surgical removal, if there's any new targets or treatments for lung nodules on the horizon.
Well, I may not be aware of every single one of those things. I know there are a lot of people working on metastatic disease in osteosarcoma amongst the COG community and amongst a lot of different research communities, some of which are probably represented on this call. But I know that that's been a major focus of a lot of organizations recently, is to fund specific research for identifying targets for metastatic disease, because that's really the crux of what the problem is, right? And so the answer is, I think things will be coming, but that there's nothing that's currently available more widely in the clinic other than aggressive metastatectomy to control those mets.
More research needed, but also more cell lines needed.
Yeah, absolutely.
I've never met a patient family or a patient who isn't willing to give body parts that we've already given, samples, et cetera, toward research. What I don't know is, and many patient families don't know, is how do you do that? Like, how do you say, please take this and send this to X place, or please use this, I'm done with it. Mm-hmm. Like we need, looking at originally, I think one of your first slides was saying you had 10 cell lines for osteosarcoma. Mm-hmm. And I'm like, oh my gosh, we need more cell lines in osteosarcoma, and fresh cell lines and fresh tissue. And do you have any idea on that? I mean, obviously we have the Osteosarcoma Project, which is moving mountains in providing researchers with data.
Yeah.
But how do you get it? How does that work?
We try to get cell lines every way we can, but there are so many people who are now, working on the best ways to make new cell lines, and so that the quality of new cell lines and the ability to generate new cell lines has really advanced within the last couple of years, and so I think that that's going to really help us incorporate them into screening projects like the Pediatric DepMap project. So we're trying to collaborate with a lot of people to get those lines into our pipeline, because again, if we put all the data together, it's just so much more powerful than a couple lines here, a couple lines there. Yeah, Ryan, do you want to say something else?
I was just going to say, I just want to make sure everyone knows that this is like a huge priority right now for us within the COG community. And there's been a lot of, there's a lot about osteosarcoma that makes that a particularly difficult thing to do because it is a bone tumor, and even more than like Ewing, right? It's a bone tumor of bone. And so a lot of our historical samples just aren't useful.
Yeah.
There are two big COG clinical trials on the near horizon. One is completely approved and just being finalized. The other one is just in the final steps of approval that will have new upline treatments. So one of those is hoping to integrate some of the tyrosine kinase inhibitors like cabozantinib that have some really great data all kind of after the fact, and we're trying to see if that will help up front if we do that in kind of preventing metastatic disease. But a priority in both of those has been making sure that we leverage that system for collecting samples that will be more useful to the research community. And Pat Grohar and I and Brian Crompton a little bit have led an effort to try and make that happen. It's required going like to the top of CTEP at NCI to change the way that we approve biology and clinical trials and things like that. But that is happening, and I hope we're going to see how it works. But I hope that we've improved that a little bit. Also, there's a lot of local efforts that are becoming very, very active. You mentioned Alejandro Sweet Cordero. There's, Phil de la Cruz in New York. There's
St.
us.
Jude.
There's the Texas program, right? And there's a lot of effort now to kind of consolidate some of those too. So Phil de la Cruz is leading an effort to we're not going to consolidate the models, but we're going to make a place, kind of a clearinghouse for models like PDXs. So just FYI, we have like 211 PDXs now if we put all of our resources together for just osteosarcoma. And so doing those kinds of things and then sharing like Alejandro did with Lily or with the Broad, some of his models to be able to run through their system. That's starting to happen, and there's a lot of activity around this. I'm hoping that all the hard work is going to pay off here in the near future, but we're trying to fix that. A lot of people have put a lot of effort into it. And we should talk, Lily, we've got a few new lines that look like they're validated and growing well from our PDXs. We can add them to the
Thank you.
Too, so.
I will defend the osteosarcoma researcher physician community, because when people use the word silos, everybody's working in silos. I'm like, not in osteosarcoma. We have the best doctors and researchers who work so beautifully together, and it just, oh my gosh, it warms my heart. And I know I'm not alone in this. And I'm so grateful for you all working together. It means so much, and it really imparts a lot of hope, and it makes us as a community go, what can we do? Tell us what to do. Like, we're here for you. What do you need? More body parts? Yes.
Nobody wants better treatments more than the patients and their families, but a really close second is those of us who take care of them as doctors. Totally. And I think we're almost as motivated as you are. I don't think we could ever say that we're equally as motivated, but we're pretty close.
I know you are. I know you are. Thank you. We're out of time, except I have a question, actually, since I have 3 super BA doctors on this call. My question is this: do you play a musical instrument?
I do.
What do you play?
I play violin.
Yes. Okay. Ryan?
I play piano.
You do. Dr. Healy?
The radio.
I'm going to wrap this up by letting you know that next week, Dr. Katie Greenzang of Dana-Farber Cancer Institute and Harvard Medical School will be on next week. Her topic, What Next? Osteosarcoma Survivorship. Really excited to have her on. She spoke at Factor 2018, I think, 2019. She was great, really excited about that one. Also wanted to add to be sure and subscribe to our YouTube channel where you can be notified of new episodes of OsteoBites, view our library of videos across MIB and all OsteoBites topics and speakers. You can also watch OsteoBites on our website under OsteoBites and listen to OsteoBites via podcast wherever you get your podcast. Wow, you guys, this was such a great session. Thank you so much, Dr. Guenther, not only for being here and spending your time and presenting, but for the research that you do every day. Really appreciate it. Our panel, thank you for being here. Amy Woodcheck, physician's assistant, Maeve Smart, our MIB president of our junior board, and Matt, our junior board member and gamer agent leader. Really appreciate you being here. And thanks, everybody. We'll see you next week.